Hello and welcome to the overview of the integration of epigenetic and transcriptomic module. First, we go through why integration is important, touching on how there have been several different applications in breast cancer research, including the possibility to create more accurate breast cancer subtypes through multi-omics or platform integration. In the first case study, we show how previous authors have used expression of bisulfites DNA methylation information to better understand the breast cancer subtype profiles of a set of patients. Here we see the experimental outline for this first case study. In this figure, we can see an example of how DNA methylation profiling was able to identify two main breast cancer tumor categories with different estrogen receptor statuses. The next figure shows how methylation clusters were identified using PV-CLUST, showing how complex and heterogeneous breast cancer tumors can be. To further the information in this case study, we've provided a worked example of how to use PV-CLUST for clustering here. Additionally, we can see in this figure how the DNA methylation profiles can also reflect the cell type composition of the tumor microenvironment. Next, this module moves into the next case study. The first figure shows the association between DNA methylation and gene expression. As you can see, the majority of the CpGs in the genes with low expression were hypermethylated, while the genes with high gene expression were hypomethylated. You can see this difference in the scatter plots here. Next, this module moves on to a more interactive portion, beginning with installing the required packages and loading them. First, we begin with an analysis for the individual CpG data, and then an analysis for the differentially methylated reads. Through extracting gene expression data, and then creating a summarized experiment object, and then creating a triplet dataset. From there, we are able to evaluate the regulatory potential of CpGs or differentially methylated reads. We can then visualize the results and provide an explanation of the output of the code. Then we move into correlation for the sequencing data from the RNA-seq and RRBS module, beginning with loading the libraries. As performed in the RRBS downstream module, we read and join all of the methylation files, then filter, normalize, and unite them. We then annotate the files and convert the methylkit object into a data frame for ease of use. We then prepare the data frame for integration and convert the feature names. Then the RNA-seq file is imported for integration and converted to a data frame. Three groups are formed from the RNA-seq data based on the expression values. And then we only need the gene names, averages, and TSS moving forward in the analysis. This code shows use of scatter plots of the mean methylation levels in each gene expression as a function of the distance from the transcription site, giving us information that is useful in the biological interpretation. In the next section of the module, we go through the functional and pathway relation. We begin with the gene set enrichment analysis-based integration to examine the gene ratio of suppressed and activated genes, connecting related gene sets and overlapping gene sets. The category net plot can be used to connect biological ideas or genes in a network, and the ridge plot is useful in investigating pathway directions. The GSEA plot can provide running enrichment scores and information about ranking of genes. We then walk through a KEGG gene set enrichment analysis and have many similar plots in the output that provide valuable biological information. Finally, we perform a motif search using an example dataset. Additionally, we have some useful links and references at the end of the module. Thank you for listening to the overview of the integration module video.